So as you all know, Parkinson's disease is a common condition, and it is classically defined by the movement disorder. So you have the resting, pill-rolling tremor, the slowness of movement, the hyperkinesia, the bradykinesia, the rigidity, so the stiffness you see in the muscles, and the postural instability, the stooped walk, and the falls. And that has been well described, and that's been well described since Parkinson first described it in 1817. I have to say that most of his descriptions were based on people that weren't actually his patients. So of the 6 patients he originally described, only 2 were his patients. The other 4 were people who just happened to be walking in the East End of London when he was walking around, and he went up and had a conversation with them. I mean, nowadays you'd probably get arrested for that sort of thing. But I mean, that's how he originally described it, and then it was subsequently, as we will hear later on, that the pathology was defined as the loss of the dopamine cells in the substantia nigra. And then Maria was the first to demonstrate that the Lewy bodies, which were described in 1912, contained alpha-synuclein. So that is a very well-described story in Parkinson's disease. It is a disorder of movement. It's associated with nigral dopamine loss, and it's associated with alpha-synuclein pathology. And everyone would agree with that. And I think it's important because nowadays people have moved so far to the other extreme that they will say that Parkinson's disease has nothing to do with dopamine, has nothing to do with the nigra, and has nothing to do with movement. But that's not strictly true. If you treat people with dopamine drugs, they do very well. And that becomes important when we come on to talk later about experimental therapies, because most of the experimental therapies work on the premise that dopamine is still vital for treating people with Parkinson's disease. But as I've said, Parkinson's disease is clearly much more than a disease entity affecting that population of cells and a movement disorder. It clearly has a whole combination of different clinical signs. So one of the interesting aspects of Parkinson's disease is, whilst we always talk about the tremor and the rigidity and the bradykinesia, some people have a lot more tremor than others. Some people have relatively little tremor. So are those actually the same disease? Do people who have tremor persistent, I would say, not tremor dominant, but tremor persistent disease, have the same disease entity? Mood problems are very common. So 40% of people with Parkinson's disease will have a major mood disorder of depression or anxiety, and that's not just in relation to being told the diagnosis, seems to be an intrinsic part of it. Cognitive deficits are very common, and I'm going to come on to talk about that. Most patients with Parkinson's disease, if you follow them long enough, will develop a dementia. So that is a pretty universal feature of the condition. And autonomic dysfunction and dysfunction of the enteric nervous system, again, is a very common feature of Parkinson's disease. So it is not just a disorder of movement, it clearly involves other domains, and as you'll hear much more eloquently than from me after this from Kelly, will be the pathology. So obviously if you have that degree of variability in clinical features, then you would expect the pathology to be much more extensive than that, and indeed that is the case. So the pathology, whilst it involves the nigral dopamine cells, is not restricted to that site. And so that would explain some of the variability that you see. But the question we sort of asked ourselves 15 years ago was that actually, if you want to try and better understand Parkinson's disease in terms of this heterogeneity, so is it one disease entity or is it several different diseases, then how can we best approach that? So we see patients who have a lot of tremor, who don't have much mood problems. We see other people who have a lot of falls, have major problems with depression, cognitive problems. Are they the same disease entity? So how can we actually try and better understand that? And so what we decided to concentrate on was we decided to have a look at cognitive deficits. And we chose cognitive deficits because, as I say, dementia is a common feature of Parkinson's disease. And once patients have reached the stage of developing a dementia, they will be put into a nursing home. It's greatly increased risk of being put into a nursing home. Once you're in a nursing home, you'll almost certainly be dead in 2 or 3 years with your Parkinson's disease dementia. So this is a major determinant of quality of life and actually life expectancy. And it's what most patients dread. So the movement side of things, terrific. If you can help them on the movement side of things, they would be very grateful. But this is the thing that they dread. That's the thing that most of us dread, is actually the dementia. So in order to try and understand this question, I thought this would be very easy, but we actually very sensibly went and got some advice from some epidemiologists. So we actually set about trying to do this in a community-based way. Now the importance of this is that what we're actually trying to do here is we're trying to gauge what Parkinson's disease is like in real life. So most studies where people study these diseases They sit in their clinic and they say, well, let's our first 100 cases, that will tell me what this disease is all about. Well, of course, that will tell you what the disease is about in the first 100 cases that make it to your clinic. So if you're sitting in a specialist centre, you will get rather atypical Parkinson's disease, because they will tend to come to you because you're a specialist centre. So what we tried to do here was we went out into the county of Cambridgeshire in the east of England, and we tried to capture every single possible case of Parkinson's disease within the county over a 2-year period. So that's actually quite challenging, because first of all you have to be able to work out how you can collect those cases, and so we managed to do it through the GPs. So in the United Kingdom, if you have a possible diagnosis of Parkinson's, so if you're in the UK and one of you had a tremor and you weren't sure what to do, and whether it was Parkinson's, you would go and see your family doctor would look at you, and then they would refer you into probably the neurologist. They occasionally can come in through other sources. So you can use the GPs as a gatekeeper to find all the cases of Parkinson's disease. Now, of course, it relies on the fact that all the GPs will send you the cases. And what we did was we'll say, we'll take anything. So you don't have to diagnose Parkinson's. If you've got someone who's a bit stiff, a bit of a shake, not quite sure what it is, send them along. So that's what we did. And then over a 2-year period in the county of Cambridgeshire, we collected about 240 cases of people with possible Parkinson's. And what we did was we used the UK PDS brain bank criteria, as they were called in those days, to diagnose Parkinson's disease, which gives you an accuracy of about 95%. It's always a problem when you start, because of course the very early stage cases is where you make the misdiagnosis. But essentially we wanted to collect all these cases over a given period of time, over a given area, using defined criteria. So over this 2-year period, we found 160 people who had Parkinson's disease using those criteria. We had 80 people who did not fulfill those criteria, but we continued to follow them up, because normally the thing that excluded them was having more than one affected family member, which takes you out of the diagnostic criteria. But I'm not sure is that helpful. And so essentially we collected everybody in a given period of time over a given area using defined criteria. We threw a whole series of tests at them, and then we followed them, and we continue to follow them to the present day. So this was the first ever community-based longitudinal study of incident Parkinson's, incident being newly diagnosed as opposed to prevalent, which are things that are already out there. And Tom Faltney was the chap who took this on, who's now at Queen Square. And this is the 10-year follow-up. As I say, we've just finished the latest follow-up, but I don't have the data on that. So what you can see from this is that at the first time that we reappraised them, we went back through the diagnostic reappraisal, because that's the time when the diagnosis is most often wrong. So in all of the big drug studies in newly diagnosed PD, about 10% of patients don't have Parkinson's disease. There's this whole entity, for those interested, called SWEDS, which is essentially people who have a tremor but don't have Parkinson's disease, but people seem to have made careers out of this sort of misdiagnostic criteria. But essentially, we re-diagnosed people, and then we followed them. So the first thing you'll sort of see is that over a period of 10 years, significant numbers of people die. So that looks pretty bad. I'll come back to that. So of our original cohort, 63 had died. 30 people had decided that they had better things to do than be pestered by us collecting bits of information. But what you can do is you can at least just collect basic information on these people. They don't want to have all of the assessments, but you can at least phone them up, check they're alive, get a little bit of information, and ask permission to have a look at their clinic letters, so you can at least get some idea of where they are with respect to their Parkinson's disease. So this is a study that we set up. As I say, we've carried on following it. The hypothesis when we began was we had no hypothesis. So it was completely hypothesis-free zone. Because if you start off saying, well, I'm going to find 3 types of Parkinson's disease, A, B, and C, you will find your 3 types of Parkinson's disease, because that's your preconceived idea. So here we said, we'll just follow and see what happens. And what we were using as our endpoint to distinguish, if you like, was when people developed a dementia. So what we found was that actually, although all those people died with Parkinson's disease, the death rate, so your mortality rate in the community with Parkinson's disease, is no different from the normal population. So as a population, if you get Parkinson's disease, you will not die any sooner than if you don't have it. So it doesn't shorten your life, which is contrary to what most people will think. And that is because the average age you get Parkinson's disease is 68. So we've repeated this study again in the same area using approximately the same approach, and we come up with exactly the same figure. So the average age gets 68. The average age in the States, when I spoke to Ira Shulson about this, is 59. So, and I don't believe it's 9 years younger that you get Parkinson's disease in the States. I think it's 9 years younger that you get into the healthcare services in the States. So this is, as I say, average age 68. It doesn't significantly shorten your life. And if you look at death certificates, which are notoriously bad for recording these things, actually Parkinson's disease contributes relatively little to those who've died. So the vast majority of people with Parkinson's disease die at the normal age you'd expect for the population and not of Parkinson's disease. So that's a bit depressing for people like me, where I'm trying to leverage funds from to do research to say I want to cure this condition, when they'll say, well, actually they're just going to die at the normal rate, and most people die of other things, so why bother? The reason is that, as you can see, your risk of getting dementia is significantly increased by possessing Parkinson's disease. So simply by having Parkinson's disease, you're 2 to 3 times more likely to develop a dementia. And of course, there are a significant proportion of people who develop Parkinson's disease before the age of 68, where it will have a significant impact on their life. But this increased incidence in dementia was clearly what we were interested in. So we've taken a population, we've defined how common Parkinson's disease is, we can tell you something about its mortality rate, and then you can also just describe what happens. So amazingly, in the in almost any era, no one knows what the natural history of Parkinson's disease in the levodopa area. So what actually happens? So this was, as I say, it doesn't shorten your life. And then on this figure up here, you can start to see some aspects of how Parkinson's disease evolves over time. So here, on this. So this is a postural instability. So for those of you who know about Parkinson's disease, it's staged by a thing called Hearn and Yahr. So Hearn and Yahr, 1 is it affects one side, 5 you're bedbound. Stage 3 is where you develop postural instability, you start falling, and that's a critical milestone. So once people start falling, they're normally on a slippery slope down on their Parkinson's disease. And you can see at 10 years, about 30% of people are not having falls, but 70% of people are. The dementia I'll come back to, but just to briefly say, here at 10 years, 40% of people with Parkinson's disease have developed a dementia within 10 years. And here, this is another group we're particularly interested in. At 10 years, about 1 in 4 patients with Parkinson's disease are doing extremely well, by which I mean that they're alive, they're not falling, and they're not demented. So there is something different about that group compared to this group up here. And then you can do things such as using the Unified Parkinson's Disease Rating Scale, which is a rating scale developed for many years to look at Parkinson's disease, which everybody loves because it gives you numbers, and it looks terribly objective, but in fact it's terribly subjective because it's how you rate the patient. But anyway, you can use this to start to see what changes you get over time, and there are aspects of your Parkinson's disease, such as your gait, often the tremor, which are not very responsive to dopamine. So you can start to plot how the UPDRS changes over time. You can extract from it elements of the UPDRS which normally don't respond to dopamine. And that, at some level, will give you an indication of what the disease process is doing, independent of the dopaminergic aspects of it. And that's important because all patients with Parkinson's get put onto dopamine drugs. So if you're trying to develop a disease-modifying drug, you've got to have some measure by which you can say, is my disease-modifying drug really affecting the disease process, independent of any symptomatic effects it may be having on the dopaminergic network. So you can start to come up with figures such as that. Now, as I said, what we were particularly interested in was the dementia. So the question we asked ourselves is, right, we've just described what we find. So in the community, you've got a 40% chance that at 10 years you'll be demented, but 60% of people are not demented. So why is it that some people develop an early dementia and some people do not? What is the difference between those? Well, if you look at clinical measures, and I'll briefly come on to this, then if you actually look at people at baseline, then there are 3 measures which are highly predictive of people developing a dementia within the first few years. So if someone comes into your clinic, newly diagnosed, first time you've ever seen them with Parkinson's disease, if they're over 72, that puts them at a greatly increased risk of developing early dementia. If you get them to list animals, So in 90 seconds, if you ask them to list animals and they cannot do more than 20, that is highly predictive. I'd say it's actually the earliest feature of the PD dementia. So now everyone will spend the next 90 seconds trying to work out how many animals they can actually produce. And if you can't draw interlocking pentagons, which I don't know if any of you... So this is on the mini mental state examination, which probably not many of you know about, but there's a thing where you do these interlocking pentagons. So this is just a very simple test, and people with PD dementia, so they can't do this when they first present. So if you can't do this, can't produce 20 animals, and you're over 72, you're in trouble. And normally what happens with people with PD dementia is this completely baffles them. So what happens is they look at this, and they cannot see its interlocking pentagons. It's a visuospatial problem, so they just stare at that, and they are completely mystified as to how those lines relate to each other. So if any of you go on to see patients in clinic, 3 simple measures. I have to say it was a bit depressing because we'd spent 5 hours assessing these patients at each time with lots of computer tests. But at the end of the day, if you can work out their age, 90 seconds for animals, interlocking pentagons, within 4 minutes, you can normally tell whether someone's going to develop an early dementia. So those were the clinical features which were predictive of people. So this is at diagnosis. So they come into clinic, you've got Parkinson's disease, work out their age, do those 2 tests. You've got a fair idea what's going to happen to them. The question is, well, why? Why do these people do it? Well, obviously age, and I'm not going to talk about age, but, and I don't suppose many people will, but that is clearly the biggest risk factor for any form of brain rot. So whatever we talk about, all of the genes, everything else, the biggest risk factor is age. And quite what age does, nobody seems quite sure. But what we decided to do was we decided that we would look at candidate genes for dementia. Now, why did we choose these genes? In part because of local interest. So there were things that we could do locally, and in part because in the literature there were some suggestions that these would be important. So we looked at these 4 genes. I'm not going to go into all of them in detail, but essentially what we were interested in here was not whether these genes give you a risk of Parkinson's disease, as John Hardy was talking about last night. I mean, we have done that, and we have contributed to that. So tau is a risk factor for getting Parkinson's disease. So we're not interested in whether these genes increase your risk of Parkinson's disease. What we're interested in is, do these genes or these variations in these genes in any way predict your clinical course? So once you've got Parkinson's disease, does your tau haplotype influence how you behave or not? That's what we're interested in. So we're interested in, do genes influence disease course? Because if they do, that informs us about what might be driving the disease process in those who have a more malignant form of disease. And if they're critical in driving the disease process, they may be good targets by which to slow down the disease. So we looked at the tau, and what we found was, and we've done this several times, was that it was predictive or influenced whether you got an early dementia. So the H1H1 tau haplotype, which is the dominant haplotype in the United Kingdom, if you possess that, it did increase your risk of Parkinson's disease. There have been many studies which have shown that. But what it did was it increased your risk of getting an early dementia. So whatever this is doing, and there'll be people here who are much better informed on this than me, whatever the H1H1 tau haplotype does, and in our post-mortem samples, it does seem to change the ratio of the 4 to 3 repeat tau in the presence of Parkinson's disease. So it seems to change the ratio. So in some way it's driving the disease process. It causes an early dementia. So that is a risk factor for a genetic factor which is influencing whether you'll get an early dementia. And just so I can sort of just come back to how this all works. So if you now just take these simple facts, so you take a group of 20 patients, so this is my study, I'm going to take 20 patients, and one of you in this room has come up with a crackerjack therapy that you think stops Parkinson's disease. That's what you think. So I'm going to say, well, how am I going to do this study? Well, look, I've got these markers. So I've got semantic fluency, which is the animal naming. I've got the pentagon copying. I've got their age, and I can do their tau haplotype. So what's the risk of them getting a dementia within 5 years? Well, this is the low-risk group, I've told you. So if you're an H2 carrier, you can do more than 20 animals, you're absolutely fine on the pentagon. That's low risk. Conversely, if you can't, you're at high risk. Now, if I just took 20 people at random, how would they behave? Well, they would behave, if you're going to use dementia as your endpoint, which would be for this disease-modifying therapy, then this is going to be hard to show. What about that group over there? Well, if you took this group and you only selected for this, which is what most people do in trials, you say, well, actually, I'm going to use dementia as my endpoint. I've got this fabulous therapy, which I think really gets into the heart of Parkinson's disease. It's a fabulous vaccine, stops it. I'm going to use dementia at my endpoint. 5 years, hey, nobody's got a dementia. How many would you predict to get a dementia at the end of 5 years? None. So your therapy won't have shown anything. So your control group would have behaved exactly the same as your therapeutic group. On the other hand, if you'd gone over here and selected for this group and done exactly the same experiment, 5-year endpoint dementia, you would predict on those 3 variables that actually 12 of your 20 patients would have developed a dementia. And if none of them have, then you probably have got a therapy that really is dealing with Parkinson's disease at its core. So simply doing these stratification can suddenly change what happens. And of course, if you mix them all up, which is the other thing that people do, then it's very hard to disentangle whether your therapy is doing anything. So that was the tau story. The second one was the GBA gene. So John briefly mentioned this last night. So this is a gene which, when we started this project, had almost nothing to do with Parkinson's disease in 2000, 2001. There was Ellen Stransky who said, I thought this had something to do with Parkinson's disease, but nobody was listening particularly to her. But this is a gene which, as you probably know, if you inherit 2 abnormal genes, you get Gaucher's disease, which is a disease which affects the liver, the spleen, and is a sort of lysosomal storage disorder, and so it presents normally in childhood. Now, it was noted in the families of Gaucher patients that there seemed a higher incidence of Parkinson's disease in the relatives than you would expect. And so subsequently people have been looking at this, and actually GBA mutations are very common as a cause of Parkinson's disease in sporadic disease. So I'm not going to talk about that, but in our population, so if you took our incident cohort, the number of those patients who had a Mendelian form of Parkinson's disease, so LRRK2 and all of that, of the 159 patients, it was 0, not 1. We did not find a single Mendelian form of the disease, but what we did find was that 10% of them had a GBA mutation. And if you follow their clinical course, so this is a beautifully blinded study because we're not biased in any way because the gene didn't even really bother us when we started. If you look at how people did as a function of known GBA mutations, so this is heterozygote, so if they're not compound heterozygote or homozygous, which give you Gaucher's disease, if you possess a known GBA mutation on one of your genes and you get Parkinson's disease, in our incident cohort, every single one of those patients had demented within 8 years. If you had one of these polymorphisms which influenced the GBA, then you sat somewhere in between the known mutations and those who are wild type. So here is another gene that is in some way influencing the disease course. In our study, these patients didn't present earlier, so there was nothing different about them when they presented, except they all went on to develop a dementia. Yes? It didn't seem to. It didn't seem. I mean, the numbers are relatively small because although it's 10%, it's with the polymorphisms and with the mutations, it's still only 12 patients or so. So both the tau haplotype and the GBA seem to, in some way, influence the disease course. So not only did it increase your risk, but once you got it, you did badly. Now, ApoE, we couldn't really decide one way or another, and COMT is a rather different story, so I'm not going to go through those other 2 genes. But what I want us to now sort of think about is you can take this information and you can start to plot what happens with Parkinson's disease as it first presents. And of course, one of the themes which will come out, I'm sure, this week is that the future of treating a lot of these diseases is to treat them very early. So don't wait until people are well established, because by that time, all the damage is done. So whilst you could say, well, you're talking here about incident cases at presentation, that is going to be the future of trying to treat these people. I mean, people will say, as I'm sure Kelly will, that there's, you can go before prodromal PD, but there are uncertain questions there about treating people who may never go on to develop the disease you're interested in. But here, what we found clinically was that the younger patient, as I said, normal semantic fluency in pentagon drawing, behaved very differently to the older patient who had those cognitive deficits. They developed an early PD dementia, and there are genes which are associated with that. So those genes in some way are driving it. The COMT polymorphism I'm not going to talk about, but I'm happy to talk about in discussion if people would like to. So that's the clinical phenotype. Those are the genetic associations. And now what we're trying to do, well, I'm not because I don't understand it, but Andrew Singleton and Matt Farrer and others are now trying to actually look at big populations to see if there are other genes that determine whether you get an early dementia. The problem with most studies is nobody records clinical data in any detail. So knowing whether someone's got a dementia or not, it's very hard. But are there other genes? That is something which is currently ongoing in big groups internationally. So that's what we found clinically. Those were the genetic associations, and then we would say, well, what's the pathology? Well, we don't know. And we don't know because we haven't really got enough brains, but what we would postulate is that what we find on one side, the younger patients, is they have a relatively benign disease. That's not to say they won't go on and get a dementia, but at least for 10 years they won't. Probably for 20 they won't, but at 30 or 40 they probably will. But there is another group of people who have a malignant form of Parkinson's disease. So once it begins and they present in your clinic, they are going to run into major problems very quickly. And they are the older patients who you can pick up clinically who have these genes which seem to be driving the disease process. So this is what we would say is there's this benign to malignant. So I personally don't think these are different diseases. I think they're the same disease running with different kinetics. And of course, we're now trying to investigate this by trying to look at sort of cellular models of this. We haven't really gone down the IPS route on this. We've gone more down the inducible neurons, and I'm sure, as Vanya will say, that was probably not the wisest thing to do. But we will persevere with the hope that we can try and demonstrate that there are intrinsic differences in the neurons derived from patients with idiopathic disease, but with these different genetic makeup. So that then sets the scene for how do you use that information to inform where you go next. So what are you actually going to do with that information? Well, at one level, it's helpful clinically. So now in clinic, when I see patients, I can tell them what I think the future holds and then how I manage them for the next year or two, because there's no point holding back in this group for your therapies for 3 or 4 or 5 years' time, because they're going to be in trouble. So you treat them much more aggressively than perhaps we used to. But for our discussion today, what it means is that it opens up the opportunity to actually try different therapeutic strategies. So this group over here, these younger patients who have no major cognitive deficits at presentation, have a relatively good prognosis in some ways. They are the perfect group to try and put their dopamine system back to normal. The group on the other side, however, are not, because their disease is more malignant, the pathology spreads out much more quickly, and so their dopamine deficit gets swamped by their non-dopaminergic pathology and all of the problems associated with that. Now, as I say, I've already sort of laboured this point, but I just want to make it again. So if you did a dopamine therapy, so if someone in this room came to me and said, I have made the perfect dopamine cell for you, from any source, let's say a stem cell source. So if Vanya comes to me and says, look, I've got this perfect cell, absolute perfect cell, could you try it in your patients? You say, fine, I'll just grab the first 10 patients that pitch up to clinic. So you take the first 10 patients, 5 are in that group, 5 are in that group. You then say, I'm going to take as my endpoint 3 years, because I want the cell to really bed down, and I'm going to ask people at 3 years whether they feel a lot better for their cell therapy. Because at the end of the day, it's whether the patient feels better with their therapy or not. Well, this group, 3 years, 5 patients, they'll say, actually, I feel pretty good. That therapy is working pretty well. I've pretty much stopped all my tablets. I'm pretty much back to normal. So I say, terrific. Those 5 patients over there, meanwhile, have started to develop falls. They've started to develop dementia. They're starting to say, well, actually, I feel absolutely rubbish. I'm actually much, much worse because I've had your therapy. I'm now actually, much, much worse off than I was 3 years ago. So they're much better. They're much worse. You take the group, because that's what you do, and the result is it doesn't work. So therefore, the reason your therapy didn't work is because it doesn't work. It's a useless therapy. And of course, it didn't work because you've mixed up your groups. And you may think that's a bit simplistic, but that, trust me, as what has happened, as you'll discover as I take you through a tour of all of the experimental therapies for Parkinson's disease, which have now ended up in exactly the same place as a consequence of failing at some level to stratify your patients. Apart from the other big point which I want to make is that experimental medicine, when you take a therapy for the first time into patients, you have absolutely no idea what you're doing. And that sounds terrifying because you think, I do this, so aren't I irresponsible by taking things into patients? Well, one of the things which hopefully will come up in the discussion is what do you actually need to know preclinically for a therapy to go to clinic? What sort of information would you like? But whatever you discover in your rat or your mouse or even your invertebrates or your non-human primate, when you come to a patient, you are going to have to make various guesses as to what to do. So if I have a cell, what dose do I give? No idea. How long do I immunosuppress them? No idea. Where do I put the cells? Well, I have some idea on that. So there are lots of things that you don't actually know. And so in a risk-averse society, people are very worried that doing these type of experiments, which they are in people, is dangerous, and they go very cautiously. And that's right at one level. But at the other end, you have to remember that any therapy into patients, you're guessing what to do. And the chances of getting it right first time round are pretty small. And so when I come on, which I will talk about now, which is cell-based therapies, you have to remember that the first heart transplants then were done by Christian Barnard. Everyone remembers him, fantastic, great breakthrough. How long did the patient survive with Christian Barnard's new heart? I think a couple of weeks. So as far as the patient was concerned, that was a completely pointless exercise. But by learning that they could technically do it, but they couldn't suppress the immune system, they moved forward, and now we have organ transplantation as routine. But the early stages of these therapies, they all failed. So you can't expect to get it right first time. And so it's very important that you try and understand what you can preclinically, knowing that when you go to the clinic, there will be things you're going to have to guess, and you're probably going to get them wrong at some level. So let's now go through what these better biological therapies are to deliver dopamine. So there are essentially a number of approaches, and I'm going to take us through these. The first is cell-based therapies. So these are the idea, so the predominant theme is that you actually replace the dopamine cells. So we have our good group of patients. We say, what are we going to do for Mr. Jones, 55, newly diagnosed with PD? It's going to do very well. Can we stop all the problems of the drugs by giving him a better therapy? So we can replace the lost nigral dopamine cells. And I'm going to talk about taking the tissue from the developing midbrain of fetuses. There's lots of other cells that have been done. None of them work. They didn't work preclinically, and amazingly, they didn't work clinically. So one thing you can say is if your preclinical therapy does not work as well as fetal midbrain, it will not work in patients. And you may think, well, that is so bleeding obvious, why do you say it? Because people don't. So spheramine, which is this therapy down here, they did a preclinical study, it didn't work. They did an open label study where they got some signal of effect which couldn't be related to what the drug was, the therapy was supposed to be doing. They had a post-mortem showing no survival. They then went on to do a double-blind placebo-controlled trial, and it didn't work. But you knew it wasn't going to work because it didn't work preclinically, so why would it work in patients? So I'm going to talk about that. There are other things which people have done. So the group in the west coast of America have managed to take stem cells and turn into GABAergic interneurons. By so doing, they then look for a disease which they could treat with GABAergic interneurons. So they've talked about using it for Parkinson's disease by transplanting them into the striatum to deal with some of the consequences of dopamine loss. They've also used them in epilepsy. I'm sure they'll use them in any number of conditions where GABAergic cells may be important. I'm not going to talk about that. And then there's other approaches, which I'll briefly mention at the end, where people have used other cells in clinical trials. So there's a trial going on at the moment in New Zealand where they're putting in neonatal pig choroid plexus into the striatum to see if that helped with Parkinson's. I'm not quite sure of the mechanism, but I'll briefly talk about that later on. So this is an approach where I'm really going to be talking about cell replacement. Can I put back the missing dopamine cells? And I forgot to say at the beginning that in Parkinson's disease, you lose about 50% of your dopamine cells when you present with your motor features. And that's only that's 250,000 on each side. So that's not many cells. So that's tractable. 250,000 dopamine cells, all I need to put that system back to normal. So that's cell replacement. The other approach is to use genes, and you can use genes essentially in three ways. One is to replace dopamine, so you have a more efficient way of delivering dopamine at the site where the pathology is, and I'll talk about that. Secondly, to put in fertilizer for dopamine cells, so some sort of growth factor, and I'll talk about that. And the third one, which was a slightly odd approach, was to try and put in a gene therapy which changed the output of the subthalamic nucleus, which is normally glutamatergic, so it drives the excitatory output it drives the inhibitory output from the basal ganglia. It's excitatory, obviously. And so what they decided to do was to try and change the glutamate phenotype of the subthalamic nucleus into an inhibitory one. I'm not entirely sure why they wanted to do that, but they did, because deep brain stimulation works just as well. The trial didn't really produce anything, and that's now consigned to history, so I'm not going to talk about that. And then the final one is actually directly putting growth factors into the brain to restore it. So this is directly infusing glial cell line derived neurotrophic factor into the brain. So I'm going to briefly walk us through these to give you some idea of how you start off with a concept, you do the preclinical work, you go to the clinic, and what happens. And you'll discover that every single one of these ends up at pretty much the same point. And most people have largely abandoned these therapies for reasons to do with impatience rather than anything else. So let's talk about fetal dopamine cells. So this is the idea, as I say, that you're going to replace the lost dopaminergic cells of Parkinson's disease by replacing them with their fetal equivalent. So this is a field that goes back to the 1970s and really derived from pioneering work done in Sweden by Anders Björklund and his team. So essentially in the 1970s, what it was demonstrated, and the early 1980s, was that if you dissected out the developing midbrain from a fetus, so in the rat it's around normally E13, E14, and you take that developing midbrain and you transplant it into the brain of an adult rat, in this case, or you can do it in mice, what you found was that the cells survived. Now we sort of say that glibly. You sort of think, well, everyone knew that. I mean, it's extraordinary when you think about it. You take out, so you know, the pregnant rat, you euthanize it, you take out the fetuses, so they already have died, take out the bit of the midbrain, turn it into some sort of gloopy soup, inject it in the brain, you sort of think, well, I mean, nothing's going to survive, and it survived. So, I mean, it's extraordinary, because people sort of just say it glibly nowadays, but it was extraordinary that that actually happened, that you could get those cells to survive. And so during the early 1980s, it was demonstrated in a whole series of experiments and a whole series of labs that you could get these cells to survive. So the first thing was that you could get them to survive. In fact, the original experiments were done not to treat Parkinson's disease. So the original experiments were done to study development. So I say this because some of you will be doing studies and you think, I've no idea quite why I'm doing this study. And everyone seems to say I have to do translation, which is what everyone bangs on about these days. You know, you've got to have clinical translation. These studies were not done for clinical translation. Anders Björklund wanted to study the normal development of the dopamine system. They had just developed new techniques, so fluorescence histochemistry for looking at dopamine fibers. So he said, I want to see how the normal dopamine system develops. I can't really do that very well in fetuses. Perhaps if I transplanted the cells in, I could follow them over time. So the original experiments were done to study development. It was only when they went and did the adrenals transplants did they suddenly think, well, this might be a better way of treating Parkinson's disease. So during the 1980s, what it was demonstrated, early part of the 1980s, was that if you took a rat model of Parkinson's disease, which is not a rat model of Parkinson's disease, it's a rat model of dopamine deficiency. So you put in the toxin, for those who are familiar with this, you put 6-hydroxydopamine into the nigrostriatal bundle on one side, you kill off the dopamine cells in an irreversible and acute fashion. That is not Parkinson's disease, never will be, never is, but never was intended to be. What it is its dopamine loss. So if you've got a therapy that replaces dopamine, it's a fabulous model. So people always criticize the models as being useless. They're useless for Parkinson's disease, so it's not useful for disease-modifying therapy. But if I've got a cell that makes dopamine, it should work in that system, and that's a perfectly good system to use. And so what they demonstrated was that if you took it from the same species, you injected it into the striatum, not the nigra, where the cells are lost, then what you found was that they actually survived long-term, which was a year in a rat. That it made connections, it made synapses, it released dopamine, and on many behaviors the animals returned to normal, but not all behaviors. So even in this simplistic model, you could not return the rat back to normal. So what this would predict is that if I take patients with Parkinson's disease and I take the group that I think are the best group to do this with, what I would anticipate was it will do as well as any dopamine therapy, but it will do nothing more. So it's not going to cure all the problems in Parkinson's disease because, as we know, and as we'll come on to here, there's pathology at other sites. But it will do as well as their dopamine therapy. And so it was on that background that people moved to clinical trials in 1987. So these were open-label studies which were undertaken by Ulla Lindvall in Sweden, and essentially the first 2 patients, they got absolutely no benefit at all from the therapy, and then they went on and did 2 more patients. And patient 4, I'm just going to talk about, because patient 4, you will see his histology, not in my talk, but it will appear, I'm sure, in the literature in the next year, because this patient has now subsequently died. But this was a patient who, in the late 1980s, so remember, no deep brain stimulation in the late 1980s, didn't exist. No one had thought about it. And so as the drugs available in the late 1980s, this patient, when taking their drug therapy, the optimal drug therapy as existed at that time, spent 2/3 of the day off, which means that essentially the drugs weren't working for, say, 8 or 12 hours. They were stuck, they were slow, they couldn't do anything. They then had a transplant of several human VMs, so these are collected from termination of pregnancies, I think it was 4. They were transplanted into one striatum, and then the patient was followed over 3 years. And following that transplantation, the first thing you'll notice is that nothing happened. So for 3 months, nothing. So if you had an endpoint at 3 months saying, does this therapy work, chuck it in the bin, think, thanks, waste of time. They waited, and of course, one of the important things to remember about these therapies is they're not drugs, so these are not going to work immediately. These cells have to survive, they have to make connections, they have to mature, you have to learn how to use them. And so 3 years after this patient had a unilateral transplant, they had gone from being off 2/3 of the day to essentially being off for none of the day. So that's a pretty impressive response. It's even more impressive when you realise they'd essentially stopped all of their medication. So they were on no drugs 3 years, so this is 13 years into their Parkinson's disease. They were on all day, and on their fluorodopa PET scan, which looks at dopamine terminals at the site of transplantation, you can see that the transplant has slowly matured, whereas on the non-grafted side, the endogenous intrinsic dopamine system has continued to degenerate. So this is a proof of principle. So this patient, for whatever reason, has done fabulously well clinically, and that correlates with their PET scan. And their post-mortem, which I've been privileged enough to see the pictures of it, is extraordinary. So this patient who died in 2012 of dementia has on the transplanted side an entirely normal looking striatum, has a transplant that's full of dopamine cells, fibres that fill up the whole striatum. On the non-grafted side, there are about 5 or 6 fibres that you can see. So it's extraordinary that this transplant has survived long term. The other extraordinary thing, which of course raises all sorts of questions for me who's thinking about taking stem cells forward, is do we need to do transplants on both sides of the brain? So why has this patient gone from being off 2/3 of the day to on all the time when they've only had transplant one side of their brain? Extraordinary. So it clearly has the bilateral output from these structures, and the plasticity is much greater than we thought. So it was on this background of these open-label studies that things moved forward quite quickly. Yeah. So I don't know the exact figures. I think the patient was around, when he had the transplant, probably 54, 55. And so when he died, he was late 70s. So he died in 2012, and the transplant was done in 88. 10 years. So it's 34 years at the time of death. So he developed a dementia, which makes the point that a lot of these patients will, but for 20 years, he was on no medication for his Parkinson's disease and looked pretty good. So then other groups followed this. So there were groups in France, there were groups in North America that undertook similar studies, and they demonstrated that there was dopamine cells in the transplant. So this was a paper that appeared in the New England Journal of Medicine in 1995. which you probably think is ancient. I always find this with the students. I always say there's a paper in 95, and they say, oh, we don't really believe that. 95? That was years ago. But that was the first demonstration of a transplant surviving in a patient who had a clinical improvement and a PET scan. So it showed that the PET scan was showing you what was there on the graft. So there was post-mortem evidence of graft survival. They also demonstrated dopamine release on PET scanning and reactivation of motor cortical areas. So these open-label studies were very informative. Then what happened was that things went slightly cockeyed because Bill Clinton was elected into the White House. Now, that wasn't really his fault, but having got there, he then freed up federal funding for studies using fetal tissue, which up to that point could not be done with federal funding, was done privately. And there was a doctor in Denver called Kurt Fried, who I always think looks like Bob Hope, sounds like Jack Nicholson, who had been going around saying that the government was behaving in a totally irresponsible fashion by not allowing this curative therapy to be used in patients with Parkinson's disease. So he raised the ante, and then NIH funded 2 double-blind, placebo-controlled trials for taking this forward. So these are trials where patients essentially either had fetal tissue or had sham surgery. They didn't actually have dead cells implanted as you would in an experimental model. But in this case, there was double-blind, placebo-controlled trials. So these began in 93. First patient ever had a graft was 87. By the time the trial started, in the published literature, I think there were 4 cases. So it was on that basis that everyone thought they knew how to do this optimally. And that optimal optimal approach led to that headline in The Guardian in 2001, which was that the miraculous cure, which of course is what the media had portrayed this, had now turned into a catastrophe. So one of the things I always say about the press is that any therapy you take to patients, or any therapy you have, only has 2 outcomes. It's either miraculous cure or it's catastrophic. Because that's what they're selling. They're selling their newspaper. So they don't say new trial in Parkinson's disease produces disappointing result, although some patients did well, because that's not going to sell you a newspaper. So this is what the public read, and then you get this perception on what it is. So this cell-based therapy is now turned into a catastrophe. So what was it? Well, it was this study by Kurt Fried. And essentially, they took 40 patients with Parkinson's disease. 20 of them were randomized to have a transplant with fetal tissue, and 20 of them were randomized to have a hole drilled in their skull, not all the way through, and pretend surgery. So nobody knew whether they'd had surgery apart from the surgeon. So the physician seeing them didn't know. The patient didn't know. At the end of 1 year, the patient was phoned up and said, do you think you're any better? That was it. That was the trial. And at the end of 1 year, they said, no, I don't feel any better, thank you very much. Now, there are all sorts of problems with that. One is that you don't know how you were a year ago. So you often, you sort of think, well, I'm not sure whether I'm any better. Secondly, if you got into this trial thinking you were going to be cured, you won't be. So you're going to think that it's failed. And thirdly, I've already shown you, it took 3 years for that patient to get back the most response out of his transplant. So a year, they might not have got the maximum response. So this failed. So this trial failed to hit its primary endpoint, which was, are you better after 1 year? Those patients who had had sham surgery were then offered surgery, so the sham arm disappeared. And so ultimately, 33 patients were grafted. So there was no significant benefit. There was some evidence that younger patients did well, but one of the worries was that 5 out of the 33 patients who were ultimately grafted developed these involuntary movements, so graft-induced dyskinesias. So when they stopped all their drugs, they had these involuntary movements. Now, anyone who takes drugs for Parkinson's disease will get these movements. L-DOPA-induced dyskinesias, everybody gets it. It doesn't stop you giving L-DOPA. But I can always get rid of L-DOPA-induced dyskinesias because I can stop your L-DOPA. You might not want it, but I can do that. Once you've got a graft-induced dyskinesia, you have a problem because I can't get the graft back. And in some of these patients, they had to go on and have deep brain stimulation, which worked perfectly well. It was just an unforeseen side effect. Now, the problem with this trial was, well, there were lots of problems with this trial, but one of the problems was that they used much less tissue than anybody else, and they only used 1 to 2 VMs per side, so that was much less than anyone else had ever used. They stored it up for a month, which affects its viability. They went through the front rather than the top, which was a very different surgical approach, and they gave no immunosuppression, because Kurt believes it's not necessary. And they did have post-mortem survival of cells, but they were suboptimal, and that probably related to a combination of those. They subsequently went on to do a 4-year follow-up on some of these patients who did much better than the 1 year. That was published in the Journal of Nuclear Medicine with an impact factor, I think, of 8.7 versus New England Journal of Medicine 53. So everyone remembers the New England Journal. Nobody remembers the 4-year follow-up paper, which came 8 years later. But essentially, this trial failed. So that was a failed trial. There was a second NIH study that was done, which produced essentially the same result. They now took 34 patients. 11 of them had no surgery, pretend surgery. 12 had a transplant with a small amount of tissue, and 11 had a transplant with a big amount of tissue. They were followed up for 2 years, and essentially at the end of that, they found that the transplants produced no significant benefit on the Unified Parkinson's Rating Scale. There's clearly a trend that the more tissue you put in, the better people do. And in fact, if you'd collapsed across the transplant group and compared the transplant group against sham surgery, they would have improved. There would have been a significant effect. But here, it failed on its primary endpoint. 13 out of the 23 patients that were grafted developed graft-induced dyskinesias, and so essentially this trial similarly failed to meet its primary endpoint with a significant side effect. So the consequence of this was that in 2003, this whole field was abandoned. This was a pointless activity because it also came on the background of Clinton leaving the White House and Bush Jr. coming in, so it's played out on a different political background, with deep brain stimulation being driven by Medtronic becoming the universal therapy for Parkinson's disease. Even by this stage, there's only been one trial of DBS. So DBS was used for 8 years before anyone did a trial to show that it actually had benefits. So this then caused big problems because everyone said, well, this is, it's had its day, it's a waste of time. But there were two problems, really. One is that the world seemed obsessed by making dopamine cells out of stem cells to treat Parkinson's disease, which seemed a strange thing to do, given you say cells don't work for Parkinson's, so why do that? So the Fox Foundation were a great one saying, we never like this, whilst putting $15 million into making dopamine cells from stem cells. So, if it doesn't work, why invest in that? And the other problem was patient 4 was a sort of what had happened in patient 4. And there were 2 patients who were subsequently grafted in Lund, who came from London, who were followed up in London. And this is one of those patients. So this is a patient of Tom Fultony at Queen Square. This is a chap who was grafted in Lund. This was taken, I think, 5 years ago, this video. He's had Parkinson's disease now for 25 years. Those are his graft-induced dyskinesias. So Parkinson's for 25 years. He's now 15 years post-transplant, and he's on no medication. So it's pretty hard to sort of say that he hasn't benefited from that therapy. And there were 2 of them who were written up, appeared in JAMA last year, who have basically got a UPDRS motor score which is less than when they presented 25 years ago. They're on no medication, and their dopamine scans are back to normal. So what we decided to do, which is, again, a sort of advertisement for, I think, people taking therapies forward, is to try and take what you've understood from these trials and try and sort of interpret it. So what do we actually discover? So one of the problems here is that people tend to entrench into a position and just shout at each other and say, transplants don't work, transplants do work. Can't we actually just engage with the data? So that's what we did. We tried to collect all the data, which proved to be an extremely difficult exercise, since one of the NIH studies would not give us the data. Eventually I had to go to NIH and get the data, and then I was given it in a form I couldn't interpret. And then when I eventually got it, I was only given minimal data because it was felt that I might be able to identify patients if I was given too much. So being open with data is very important because it allows you to understand things. So this is a plot of all the changes in the UPDRS in all of the big trials that have taken place. So this is an improvement of more than 50%. That's getting worse by 50%. So what you can see is that the scatter is enormous. So that some patients do extremely well, as I've shown you. Some do not do well at all. It was a very safe procedure. But there's clearly, whilst a significant number of people don't, there are a significant number of people who do. So is there something we can learn about those patients which we could then use to inform a subsequent trial? And I'll come briefly on to discuss that. Now, of course, in 2008, and I'm sure we'll hear more about this, is that some of these patients who had the transplant, who subsequently died, what they found in the transplants was not only that the transplants had survived, and of course the vast majority of these dopamine cells in these transplants look perfectly healthy, but a minority of them were found to have Lewy body pathology within them, which is a bit unexpected, given that those dopamine cells in those transplants are at most 15 years old. And 15-year-old people don't tend to get Parkinson's disease, and if they do, they get Parkinson's disease, which doesn't have Lewy body pathology. So that raised all sorts of questions, which I'll briefly come back to at the end. But for, of course, the critics, this is terrific. We've got 2 double-blind placebo-controlled trials that don't work. You get side effects. And now the very thing you're treating is killing off your transplant. Well, if it is killing off your transplant, it ain't doing it very well, because that last patient presumably has pathology in his transplant, but he seems to be doing very well from it. So whilst there is pathology in it, that in itself is not a reason for stopping. And the other thing which people talk about is that you have to do these double-blind placebo-controlled trials because there is a massive placebo effect in Parkinson's disease. Now, at one level that's true, but some of the people who are the biggest proponents of this were involved in the second NIH study, where they demonstrated quite elegantly that those patients who actually didn't have a transplant but didn't know whether they had it actually got worse. So the placebo effect isn't as big as people say, and it's certainly not there in all trials. So the need to do these double-blind placebo-controlled trials, I think, is questioned. So using all the information that we collected on all of these patients over a number of years, we put together a new grant. So we collected everyone together, and we wrote a grant, Trans-Euro, which brought together all of these groups in Europe, which is currently ongoing. And essentially what we've done in this study is that we've taken those characteristics of patients which we think predicts that they will do well. So that's that group I had on one side of the slide. So they're the younger patients, less advanced, without really any L-DOPA-induced dyskinesias. We've optimized how to prepare the tissue because one of the reasons you might get graft-induced dyskinesias relates to contaminating 5-HT neurons in the transplant. And the other question which people raised about graft-induced dyskinesias was you were producing dopamine hotspots by the way you were delivering the tissue into the striatum. So we've got a technique by which you can evenly distribute the cells over that structure. So we've sort of defined what we think are the optimal way as best we can with this. We've taken a design where we take 150 patients with Parkinson's disease and we randomly select 20 of them to have a transplant so we can compare our transplant against well-matched controls, and all the patients wear baseball caps, not because we want them to be trendy, but so that we can video them and then send them to a third party who can then rate them independent of whether they had a transplant or not. So that's how we've designed the study. We have a 3-year endpoint at this stage. That is our primary outcome measure. As I say, they're blinded video ratings, and we're currently here, and we actually grafted our first patient 3 weeks ago, 9 years after I started this process of trying to get this back into the clinic, which gives you some idea of how long it takes. And when you hear Alistair Compston talk at the end of this week about his new therapy, you'll hear a similar story of how long it takes from an idea to the first trial to actually getting something which is a useful therapy. Now, despite all of this, the problem with this whole approach is this is using fetal tissue. There's a whole series of ethical issues with that. The biggest problem is, of course, just logistics. So for every single one of these patients, I have to get at least 3 to 4 VMs per side. So that first patient we grafted 3 weeks ago, that was our 5th attempt to graft them, because the previous 4 attempts had failed because we didn't have enough tissue. So this has always been seen as a stepping stone, this trial, into stem cell-based therapies. And what stem cell-based therapies are we thinking about? Well, I'm not going to labour this point because various people have different views on it, but I would essentially say there are only 2 major sources of cells which are likely to go to clinic, or that's what people are talking about. One of those derived from embryonic stem cells, so the spare embryos from IVF programs, from the established cell lines which are already there. So we're not making new ES cell lines, we're using the ones that are already there. And then, of course, the iPS technology that I'm sure we'll hear more about from this afternoon. So these are the 2 therapies that we think will go to clinic first. I think all of the other stem cell therapies that people talk about for Parkinson's have almost no preclinical data to support them. Of course, if you want a stem cell therapy for Parkinson's, there are at least 400, 500 clinics around the world you can go to tomorrow to have it, but none of them have any basis. So why do I say these cells? Well, I think the human embryonic stem cells now have been demonstrated, really through the work of Lawrence Studer and more recently Marlin Palmer to make authentic nigral dopamine cells without tumour formation. So Lawrence had a paper in Nature in 2011, and then this paper in Cell Stem Cell last year from Marlin demonstrated that if you took human ES cells, this is from the H9 line, and you turn them into dopamine cells using their particular protocol, you could get cell survival, no overgrowth of cells, and not only could you get authentic nigral dopamine cells as much as one can define that, but also, which was different to what had happened with the Studer cells, was you got fibre outgrowth, which of course is vital, because if these cells don't innervate the striatum, you won't have a functional graft. So we are now taking these cells forward into the clinic for Parkinson's disease, and we're just starting that process. Now, of course, we're doing that. Lawrence Studer has a grant to do that. But the alternative to get around some of the ethical problems with ES cells and also some of the issues to do perhaps with immunogenicity is to actually obviously use iPS cells. And so there are various studies which have demonstrated this. Iron cells I'm not going to talk about. But the question then arises as to whether you'd really want to use iPS cells derived from the patient themselves because of the issues to do with pathology. I've already demonstrated that unrelated fetal tissue acquires the pathology of Parkinson's disease. What would happen if you put dopamine cells derived from the skin or the blood of patients who already had Parkinson's disease? We don't know. So there are issues to do with autografting. There is a more practical problem that this is just not feasible. So to take biopsies from every patient, grow up their iPS cell line, select the one you want, turn it into dopamine cells, quality control it, do it all to regulatory standards for a therapy that will benefit one person will never be a therapy. So one way around it is to make up banks of people who are HLA matched, and that's what they're doing in Japan. So stem cell therapies are moving forward, and learning from what we did with fetal tissue, we've tried to do this as an international consortium. So we set up a group a couple of years ago, which has the rather grand name of G-Force, which brings together the major groups around the globe who work on this within Japan, North America, and Europe. And essentially, the sort of headline on that is that that will move forward. So the Japanese are going to go with iPS-derived cells from the peripheral blood of patients with Parkinson's disease. And they hope to do a trial next year or the year after. The group of Lawrence Studer are aiming for 2018, and we are thinking of going about 2018 and 19. So in the next 5 years, I imagine there will be some stem cell-derived dopamine transplants taking place in Parkinson's disease. Now, I've gone on a bit long, so I'm going to try and speed up for these last bits to just take you through some of the other therapies that are out there. So what about gene therapies? Well, this is a gene therapy we were involved with in Cambridge, mainly came out of Paris. This is just to sort of run through some of the other therapies that are out there. This is a gene therapy where basically patients are given a gene which makes dopamine. So it's a tricistronic vector which has all the synthetic enzymes for making dopamine. It's injected into the striatum. They did a study of 15 patients, open label, dose escalation. And what they found was that this was an extremely safe therapy. There were no side effects, and that there was a signal of efficacy. So this is a therapy which has shown something which is worth taking forward. Whether this is better than any of the other dopamine-based therapies is unclear, because this isn't repairing the brain, this is just delivering dopamine more efficiently, but they're going forward with a trial next year. They have funding for that. And then I'm just going to briefly talk about growth factors to show how you can go wrong again in quite quick fashion. So the idea here is essentially you're giving a fertilizer for dopamine cells. So GGNF, which was discovered in 1993, went to a clinical trial in 1998 where it was injected into the ventricles of patients with Parkinson's disease. It demonstrated that it had no efficacy whatsoever, which is what you would expect, because if you put it into a fluid-filled cavity, it will go around a fluid-filled cavity and it will come out of the fluid-filled cavity and it will never get into the brain. So that demonstrated that it didn't work if you put it into the wrong compartment. Steve Gill in Bristol then decided to inject it directly into the striatum of patients with Parkinson's disease. So you inject it in and you try and rescue the dopamine system, and a group in Kentucky did exactly the same. So there were 15 patients who were given this therapy initially, 5 in Bristol, 10 in Kentucky. Steve Gill's paper showed that these patients improved. So here's the UPDRS over here. When they've stopped their medication overnight, what you find is it improves with GGNF. So they clinically improved. There was an improvement on their imaging. One of the patients died 18 months afterwards from a clot to the lung unrelated to the Parkinson's disease. and they discovered there was TH dopamine fiber sprouting around the infusion site in that patient. And one of the long-term follow-up on those patients has showed a stabilization of disease once the therapy was stopped after a number of years. So these open-label studies were very encouraging. It still wasn't clear quite how to do it, but as you might imagine, what happened was that it went to a double-blind, placebo-controlled trial. It was sponsored by Amgen, who are the people who made GDNF. So this was done on the basis of 15 patients. They did a 34-patient study. 17 had the active ingredient, 17 did not. It was delivered at a different dose in a different system down a different catheter, so it had no resemblance to the original trial. And you'll be amazed to discover it produced no effect. And so some people will say, again, it just shows there's a massive placebo effect in Parkinson's disease, which is why you need a control group. What this elegantly demonstrates is there was no effect. There was no placebo effect because there was no effect. So it didn't work. There was something on the PET, but essentially it was abandoned. Amgen took it off the market. Everyone said GDNF doesn't work. Open label studies are very misleading. But as I've already said, it may not have worked because essentially there were a whole series of parameters which were not the same as the open label studies. So they didn't select for patients in any particular way. They delivered it in a way that hadn't been tried before, and there were problems with the trial design. So this failed. Amgen took it off the market. Everyone had to stop it. Caused a huge hoo-ha. But they have now, Amgen have released GDNF. It's gone to another company. And so there's a trial which is fully recruited and ongoing in Bristol at the moment where they're delivering GDNF into the brain. A similar therapy was done with a gene therapy called Nurturin. They couldn't make a gene therapy with GDNF because GDNF was owned by Amgen. So they had to go for son of GDNF, which was Nurturin, which was less potent. They demonstrated it worked in animal models. They went to a double-blind, placebo-controlled trial where they demonstrated that there was no benefit at their primary endpoint, which was 12 months up here. But when they went to 18 months, they started to see a separation. And of course, you are giving a growth factor here. So you are trying to repair a system, so you're not expecting things to happen very quickly. They then went on to do a further study where they injected it into the nigra and the striatum of patients with Parkinson's disease, and it didn't work. And as far as I know, they've never published this paper. So this has also gone the way of other therapies. Now, why has this not worked? Well, one of the reasons, I think, relates to when they used it. So essentially, all of these therapies, especially these growth factor therapies, have been given in patients with moderately advanced Parkinson's disease. And of course, you'd be much better off using it much earlier on. And the reason for that is if you look at the dopamine fiber innervation of the striatum over time, these are slides that Jeff Cordua gave me, that by the time you're infusing GDNF into the brains of these patients, they essentially have no dopamine fibers left to rescue. So however good your fertilizer is, if there's no seed, it ain't going to work. So one of the problems is whether they went in too advanced stages. It may be the fibers there, they just down-regulated, but certainly it's probably not the optimal time because you've lost so many of the fibers. The second is you don't know, which comes back to a point I made earlier, how much to give. So this is the volume of distribution of Nurturin in a brain that was treated. So this here is the human putamen, this whole structure, and you can see that the infusion has only covered a tiny fraction of it. So the drug has almost certainly failed because they gave the wrong dose in the wrong stage patient. And so that is why, and probably delivered in the wrong way. So trials fail for those reasons. So what have we discovered up to now? Well, I would say that fetal cells work with some benefit are inconsistent, but we're moving forward and stem cells will be there. Gene therapies are moving forward. The growth factors, I think, are still unproven, and so there are trials that are moving forward with that. So what is planned is just to sort of recapitulate. Transuro is obviously ongoing. I've talked about the stem cell trials, and this pig choroid plexus cell transplant trial will release its results next Monday at the movement disorder meeting. Gene therapies I've already talked about, but they're all coming back to clinic. So whereas you may think this was all yesterday's therapy, they are all actually coming back with people being a bit more critical about what's gone on in the past, and as I say, the GDNF study. Now I'm going to briefly just run through these last bits just to illustrate some of the other ways in which people are thinking of moving forward on those patients who don't do well. So these are patients who are going to develop an early dementia. So these are disease-modifying therapies. I'm in the home straight now. So this is the patients, who are going to develop the early dementia. What can we use? Well, those therapies aren't going to help them. So I'm just going to flag these up because, of course, most of you in this room will be working around novel therapies to try and treat neurodegenerative diseases. Well, one thing you can start by doing, which is what a lot of people are now doing, is saying, well, why don't we do drug screens with drugs which are already out there? So there may be a whole batch of drugs which are used for other things which could be useful for Parkinson's disease. So there's a thing which was set up by the Cure Parkinson's Trust called Link Clinical Trials, which does that, just that. Are there drugs being used for other diseases which might be useful for Parkinson's disease? And the drug which they've had the greatest success with so far, that's only been going on for about 3 years, is exenatide. So exenatide is a GLP-1 agonist which is used for diabetes and has preclinical evidence that it might work in Parkinson's disease. So Tom Faltney, who props up all the way through this talk, did a study in London where he took patients with Parkinson's disease. He injected them with exenatide, I think twice daily, over a 12-month period, and recorded what happened to them. And he took a group of people who were equally disabled with their Parkinson's disease, didn't give them placebo, just followed them up. And what he found was that over a 12-month period, those who had exenatide seemed to actually improve. It's a GLP-1 agonist. So there's a whole group of these drugs which are now used a lot in diabetes, which are thought to stabilize blood glucose levels. So the advantage of it is you don't tend to get a hypo with it. So it just stabilizes what you've got. Now, how it works preclinically has been a bit uncertain. So people talk about neurogenesis. Well, I have no idea what that means in this system. But there's clearly some evidence that it may work to promote dopamine recovery. And the advantage of these is that there are a lot of them out there, and some of them you can give twice daily, some once daily, some once a week, some once a month. So they actually are very long-acting, and so you can see that they could be quite useful, and they've got quite a safe profile. And so what Tom found using the UPDRS in those that actually had given this drug, they actually improved. He then stopped at 12 months and followed them up for 2 months after that, and then for a year. The idea being, if this was all placebo effect, because it was an open-label study, that if it was placebo effect, you'd expect them all to go back to where they were once you'd stopped the drug. And the reason he had to do an open-label study was he had very limited money, and to make a placebo-injectable device, which is what you needed for this, would cost £250,000 to £400,000. So that seems like an extortionate amount of money for something where you're just trying to see a signal. So not only did he see an improvement on the UPDRS motor, but he also did the Mattis Dementia Rating Scale, so a cognitive measure, and found exactly the same, which was sustained at 14 months and at 24 months, i.e. 12 months off the therapy. So this has now led to a Fox-funded, Cure Parkinson's-funded phase 2 study to look at exenatide in people with Parkinson's disease to see whether it's disease-modifying with some imaging. And this has led to a whole series of new trials with drugs which are already out there. So as I say, there are a lot of these GLP-1 agonists. Simvastatin, which you'll all know is used for cholesterol, has some anti-inflammatory effects, may work in preclinical models, is going to a clinical trial in Parkinson's disease. Isradipine, which is a calcium antagonist, doesn't seem, well, I'll probably be careful what I say. I mean, there's a $23 million trial going on with this. I mean, it didn't work in the phase 2 study, so I don't think it'll work in the phase 3 study. But anyway, it's a nice idea. That's an antihypertensive. Metformin, which you'll know is used for diabetes, polycystic ovaries, is going for a trial. Deferapane, which is an iron chelator used for people with iron overload, is also going to clinical trial, and EPI-743 is a drug which is used to treat mitochondrial disease, is going to clinical trials. So these are all drugs which are out there for other uses, but are going to clinical trials. So this is a way that people are thinking, you know, you've got your model system, chuck everything that's got a license for use in clinic, does it work? And you can go straight to patients. Of course, there are assumptions that the dose is right, it gets into the brain and all sorts of other issues. So that's one approach. This approach I'm not going to talk about. This is where you can take some novel approach. This is something we've been doing with a non-coding RNA which binds to complex 1 of mitochondria and seems to be a very good way of protecting cells, which I'm happy to talk about afterwards. But the last thing I wanted to talk about, which I think in some ways sets us up for the next series of talks, which is novel approaches based on novel insights. And the reason I want to flag this up is this shows you how a field can gain a momentum very quickly and how one novel therapy can inform you about a novel approach. So this goes back to what I was saying about 3 hours ago about transplants acquiring the pathology of Parkinson's disease. So why have these transplants developed Lewy body pathology? Well, there's a number of possible theories. One is that the inflammation which you see around the transplant promotes in some way alpha-synuclein aggregation into Lewy bodies. But the thing which has gained the most attraction is the idea that the alpha-synuclein pathology you see in the transplant has been transported into it, if you like, from the host brain. So that the alpha-synuclein, whatever goes wrong in Parkinson's disease, the alpha-synuclein in some way changes its conformation, whatever, it then templates pathology in the endogenous system, much the same way as prion proteins do, and then you actually seed pathology and start a pathological process. Now, there are big differences between this. So prion protein, so it's not really a prion disease, it's alpha-synuclein behaving in a prion-like behavior. And I say that because prion disease, for those who know it, this is where you spontaneously get a conformational change in your prion protein. So it becomes prion protein scrapie, and that then seeds, templates the normal prion protein to take on the abnormal one. And clinically what you see in these patients is they start and they accelerate away, which is not something you see in Parkinson's disease. But the idea here is that alpha-synuclein in some way is spreading from cell to cell, triggering a pathological process. So Maria had this very nice paper, not done in the UK, because I don't think the Home Office would have allowed us to do this particular study, where she chronically fed mice rotenone. So for those who don't know, rotenone is a pesticide which has been shown to induce a Parkinsonian-like pathology in animals when injected into the brain. In this case, that the animals were chronically fed it, and what they found was that the pathology, alpha-synuclein pathology, began in the gut, then it was found in the lower brainstem, and ultimately found in the nigral dopamine cells. So this then raised the question about, perhaps the pathology begins in the gut and then spreads up, and is there pathology in the gut in patients with Parkinson's disease? Well, it's known that people who have Parkinson's disease have alpha-synuclein pathology in the gut, although there's some questions about the antibodies that are used. And then Jeff Cordua had this paper where he looked at patients who had Parkinson's disease, but for some other reason had had a bowel biopsy done some years previously. They then stained those bowel biopsies to show they had alpha-synuclein pathology in the bowel ahead of getting Parkinson's disease. So then various people have worked on this. Virginia Lee, most notably with Patrick Brundin, have then developed a whole industry around injecting aspects of alpha-synuclein into all sorts of different model systems to demonstrate that alpha-synuclein can spread and that it may be actually able to trigger a pathology. And people are now looking at extract of brain to see whether extract of human brain, see if it behaves in a similar way. I'm not going to talk about this because all I want to do is sort of get across a concept, which is that this experimental study with cell-based therapies produced a result that no one expected. Looking at that opened up a whole new possibility, coupled to the discovery that alpha-synuclein lay at the heart of Parkinson's disease, so you could develop an antibody to stain for it, based on the pioneering work we're going to hear about later on today about how you stage the pathology of it, and suddenly you can start to put together a whole new theory about Parkinson's disease, that it may be you have a genetic predisposition. I've already said that, John Hardy said it last night, but there may be something in the environment that gets into your nervous system, whether it be your enteric nervous system or your olfactory bulb. Once it gets there, it starts a process that's going to carry on. So whatever happens, the alpha-synuclein in your gut does something, it changes. It then templates the pathology, which then spreads up to the brain. Once it gets into the brain, it spreads throughout the brain. So this is a whole new idea about how Parkinson's disease comes about. Now, you may sort of think, well, so what? But the thing is, this has sort of gained a huge amount of excitement. There are probably more reviews on this than there are scientific papers. But it has raised the possibility that if alpha-synuclein changes in some way and spreads in some way, can we not immunize people to kill off whatever that alpha-synuclein species is that's bad for you? So this began a number of years ago, and there are various experimental papers to do this. This is now in the clinic. So there was a meeting in Innsbruck that Maria and I were at in early part of this year, where they presented the results on 30 patients who've had immunization therapy for Parkinson's disease. I'm not entirely sure what they've immunized against, because it's a company and I'm not quite sure what they've done. But basically they took 12 patients, gave them a high dose, 12 patients a low dose, 6 patients nothing. They then looked at the antibody response in the periphery, and in those who mounted an antibody response, they seemed to stabilize their disease. So this then raised a whole new idea that if this is the problem with Parkinson's disease, should we be looking for people before they ever get Parkinson's disease, this prodromal premotor, and immunise them? Or should we just immunise everybody over 55 and stop them getting this disease? Now, my anxiety with this field is it has gained a momentum which is rather similar to that which happened with cell-based therapies 30 years ago. And so the worry is that it will rush forward with huge speed, it won't work for some reason, and everyone will abandon it, when again, it may work, you may just have chosen the wrong patients and the wrong trial design. So what I've tried to do in this talk, which I've gone on far too long actually, is I've tried to show that you can stratify patients using relatively simple measures by following people over time, and that gives you a much better idea of how the disease behaves in different groups, which may allow you to design better trials. There's this whole idea of prodromal PD and the idea of treating earlier, which I haven't really talked about, but I think going earlier, at the moment we can only really do incident cases when they first present, but it may be we could shift back with that given some of the data I briefly mentioned at the end. There's a whole new line of dopamine therapies. So for those of you who think cell-based therapies had their day, it's all DBS, it's all dopamine infusion therapies. I think there is mileage in cell-based therapies, and there's a lot of going on the gene therapy front. And then there's a whole new range of disease-modifying therapies which are out there going to clinic. So I think there's a lot of excitement in the world of Parkinson's disease that there's lots of things that can be done. No one of them is the best way to do it, but there are various new therapies that people are thinking about, and it's useful to think in your own particular disease area, all of the principles I've described here can apply to whatever disease you're studying. And so there are various different strategies you can think about. And then finally, there's just lots of people who've actually helped me over the years doing all of this work. Particularly, I'd like to thank all of these people down here, because it's very easy for me to stand up here and say, you know, I set up this cell-based therapy, I did this. But actually it's not. This is collaborative work. And so the one thing I would encourage everyone to do is, if you're working on novel therapies and you're thinking of going to clinic, get together with all the groups that do it, because you're much better working together than trying to go for gold and be the first one to finish to get over the finishing line, because you'll also almost certainly be the first one to fall down when it all starts to go wrong. So I will finish there, and happy to take any questions or discuss any aspect of this.